record in here. All right, so here's week 7 in this class. And that pretty much means we're almost halfway through with the semester already. Pretty crazy, right? This is actually the last week of quad 1 courses. So this is the last week of the first half of the semester, and I'll be moving into the second half. So actually, let me start passing this sign-in sheet around while I'm remembering it. Okay, so lab this week, we'll get a break from the dissection. So you'll be taking a quiz on the dissection stuff, but we're not actually doing dissection in lab this week. Okay, the lab is over cardiovascular physiology. Okay, so it's a different handout that you need to have ready this week. Okay, so it's links right there on Canvas, so make sure you have that handout available. Okay, and yeah, so cardiovascular physiology, but the quiz this week in lab is a bigger quiz. Okay, so this one is a 14-point quiz. The one next week, well, I don't know if it's next week, but there's also a 16-point. Yeah, next week is cardiovascular physiology, but then the one after that is a 16-point quiz. Okay, it's just because we start getting more and more information, and we want, really these quizzes are to kind of keep you up on the material to help prepare you for the practical when that comes. So there's just more and more information, so a little bit more point value to these two quizzes. So the things that the quiz is on is the organs of both the fetal pig and the human. Okay, let me just go to the handout. I've got it here to make sure we're on the same page, literally, of which page I'm talking about. Okay, so here's our handout. Okay, so we've done that stuff. There will be some review material that's on this. Okay, that's on the veins of the human again, and those coronary blood vessels again. These guys right here. Okay, you can't see where I'm pointing right there. Okay, those guys. So coronary blood vessels and these veins, that'll be kind of the review part. Okay, and then the organs of the fetal pig. Okay, so those start here. Okay, so these bolded terms here are the organs that you need to know. Okay, so all the way down here. Okay, ending down there for the fetal pig organs. Okay, and then we have arteries of the human upper body. Okay, so those are listed here. For the ones that have something underneath them like this that say what they supply, you need to know that too. Okay, so one question might be, what is this? You know, pointing to the artery, what is this? And then the second one would be, what tissues does this supply? Okay, but for the ones that don't have anything listed underneath them, you don't need to know what it supplies. Okay, it's just those. There's really only 8 here so far that you need to know that for. Okay, and then you need to know these organs in the human as well. Okay, and then yeah, I believe that is it, right? So we got the organs of the fetal pig and the human, upper body, human arteries, and the review part is those heart blood vessels, those coronary blood vessels, and the veins. Okay, so that's the lab quiz for this week. All right, and just like the other labs where you do like a worksheet, you're going to do part of it in lab, and then you're going to complete this part here, the online portion, by Friday at 5 p.m. Okay, we had a kind of a small flood over in the Rohr Science Building over the weekend, and because of that, we're having to shift a couple labs around. And so those of you that have the 5:30 lab on Thursday, okay, so if you're in lab section 2 or 3C, 5:30 with Dr. Silva, that one will actually meet in it's going to meet in SATER 105 instead. So it's on this side. It's the microbiome lab. If you guys know where that one is, it's just right there. It's the one that faces the entrance when you come in right here. Okay, so it's just on the other side of the building, right over there. So SATER 105. So you guys will be in there instead of 117 this week. Okay, because there was a class that was supposed to meet in one in Rohr that's not going to be able to meet there. So they're going to meet in the other one because they need to use the cadavers, and that's why we're shifting things around. Okay, so if you're in that Thursday night lab, that you're over in that one. Okay, what else? Let's see. Yeah, so then this week in class, we'll have our third TBL day on Friday. So this is over blood typing. So it's like the type A, type B, O negative, all those things. Okay, that as well as the blood clotting process. Okay, so make sure you watch that video, study that hard for the IRAT and TRAT on Friday. All right, and yeah, and then lecture quiz 5. And let's see if I said in here what lecture quiz 5 is over. Yes, I did. So 2 things: blood flow and pressure, and blood composition. Okay, you know what that is? Is that right? Yeah, that is not right. yeah, I haven't finished that quiz yet. So I'll let you know after class what that one's going to be over, because we haven't done, you won't have done the composition one yet. So yeah, I'll get back to that on after class, and let's send out an announcement to verify that. But yeah, that quiz isn't totally done yet, so that's why I don't have that up to date. Okay, but I'll get that done so you can get that. I'll get that done probably by tomorrow, so that way you can do that lecture quiz earlier if you'd like. Okay. All right. So any questions about what's going on this week, guys? Either on Zoom or here in the class? Okay. All right. I wanted to do one brief thing with exam 2 here. Okay. Let me pull up my notes here. So there's just a few things I wanted to touch on for exam 2. This is kind of my way of going over the exams. Again, if you want to meet with me individually to have me kind of walk through the questions that you missed with you, I'm happy to do that. You can sign up for a office hours appointment for that. My calendar right now, if you go to sign up, it'll just say spring advising appointments. You can take one of those slots. That's fine. I'm doing all my advising right now too, but that's you can sign up for one of those. Doesn't matter. But these here and these are the kind of things that I wanted to talk about, because these were the ones that more students missed on the exam. So obviously this is kind of helping you prepare for the final, where it is cumulative and some of the stuff can come back. Okay. All right. So the first thing, the whole renin, angiotensin, aldosterone system. Okay. A good chunk of students miss the part about what leads to the release of renin. Okay. So renin is released by the kidneys in response to low blood pressure being sensed by the kidneys. Okay. So low blood pressure in the kidneys leads to renin release. And that renin release eventually leads to the formation of angiotensin 2, okay, which is, I was trying to find my pointer. I don't think I have it here. Let's do it this way. Yeah, angiotensin 2, right? So there was one kind of couple part question that a lot of students missed on that one, okay? And then angiotensin 2 is the one that has all kinds of drastic effects on blood pressure by vasoconstricting, by causing aldosterone release, by causing ADH release, by causing sympathetic nervous system activity. Okay, another one that any of them that you guys that the class, they're like less than 70% of the class missed. Those are the less than 70% of the class got right, are the ones that I'm highlighting here. Okay, so the other one was cortisol release is regulated by the pineal gland, right? That's that little tiny gland right in the middle of our brain, and that is where melatonin is released from. Okay, and that melatonin is released in response to how much light your retina receives. Okay, so that was one a lot of people missed. There was definitely some confusion around this part of the sympathetic nervous system, right? So the sympathetic nervous system is typically going to be a preganglionic neuron going to that ganglion, and then a postganglionic neuron going out to a target tissue. Okay, but then there's this kind of other branch to it that is a preganglionic neuron going to the adrenal medulla, right? That little gland on top of the kidneys. Okay. And then from there, the adrenal medulla releases these two hormones, epinephrine and norepinephrine, okay? And then those are going to go through the blood and bind to any receptors that they find that work for them, which are always going to be adrenergic receptors, okay? So there was there were some other options there in that question, like muscarinic receptors or the cholinergic receptors. Okay, but when it's the target tissue in the sympathetic nervous system, it's always adrenergic receptors. Okay. There was that question about atropine. That's that drug that the optometrist uses to dilate your pupils. Okay. So the parasympathetic nervous system is always going to be cholinergic receptors. Okay, because it's always acetylcholine as the neurotransmitter in the parasympathetic nervous system. And one of the question or one of the answer options was like muscarinic adrenergic, I think, and those never go together, right? Maybe a quarter of you chose that one, but those ones don't go together, right? Adrenergic might go with beta or alpha, but it never goes with muscarinic or nicotinic. Okay, then muscarinic or nicotinic only goes with the cholinergic ones. Okay. All right, so the whole thing on the menstrual cycle, the first one that a lot of students were confused on was in the proliferative phase, estrogen levels are rising, right? They're kind of starting to rise, and that's providing negative feedback back up to the pituitary gland and hypothalamus. Okay, and so there was some confusion on the negative or positive feedback part there. Also some confusion on whether it was estrogen or not. I purposely didn't have inhibin as one of the answers in that question because that one also provides negative feedback, but that wasn't in the answer bank there. Okay. And then yeah, for the release of LH and FSH. So a lot of students just chose one LH or FSH, but it's both, right? Those both get released together by the pituitary gland. Okay, and then this one, this is always an important concept, and I always want to make sure that you understand that LH is the one that causes ovulation, right? So that at some point we get enough estrogen released where that switches to positive feedback, right? And then we get positive feedback, so then we get a lot of LH release. We get a lot of FSH release too, but the question specifically said choose one hormone, the one that leads to ovulation, the one that leads to the rupture of the follicle, right? So that one's got to be LH. And a lot of students didn't choose LH there. Okay, so luteinizing hormone, right? It gets that name by turning the follicle into the corpus luteum. That's why it's called that. Causes ovulation. Okay, and then the last one, there was that one picture of the heart electrical conduction system, and the third one was like which of these send signals faster than the rest of them or something, right? That should have been the Purkinje fibers, which is the very bottom part. Okay, there was only about 30% of you got that one right, so that was why most of you saw a point go back, or a point go back on your chest. If you were wondering why that was, I just threw that one out. And honestly, instead of going through and seeing who got it right and wrong, I just gave everybody a 1.5. So, so I didn't feel like going through every single person's test. So everybody got a 1.5 there for that one. Okay, so that was pretty much the ones that I wanted to go over there. So, any questions in general on the exam there? Or, yeah, anything on the chat or on Zoom? Got one on the chat here with limits. Okay. All right. Cool. Okay. Well, again, if you want to go over your test specifically, happy to do that with you over Zoom. Oh yeah, just sign up for one of those appointments. Okay. All right. So let me go back to the beginning of this one here. So today we're continuing on with the cardiovascular system, but we're going to move into the blood vessels now. Okay, we've worked with the heart for a few classes now, and now we'll go to where the blood goes next. After it gets pumped out of the heart, it's got to go to those blood vessels to get distributed through the body. Okay, have any of you ever been to one of those bodies exhibits, or there's a bunch of different kinds, but yeah, a few of you had. So maybe you saw this model when you were there, but they have full human bodies that are just blood vessels usually at those exhibits, and they're pretty spectacular to see. That's what this is. Nothing else there, no bones, no skin, no anything, just blood vessels, right? And it's just pretty remarkable to see the complexity of the vascular system in a human when it's all by itself like that. So let's go ahead and do this question. You can take a guess here. So if you laid out all of those blood vessels, all the ones on that previous slide, all in a single file row in your body, how long would it be? What do you think? Take a guess, obviously. All right, give you about another 10 seconds. You can just take a guess if you don't have a clue. And this was actually done in a scientific paper. They kind of tried to extrapolate these the numbers here and figure this out. And so yeah, if let's see what you guys said here. Yeah, so kind of a normal distribution there around D, but surprisingly, it's this one here. Yeah, 60,000 miles. And so 99% of that is capillaries, which are microscopic, right? One red blood cell can fit through at a time in a capillary. Okay, so they're very, very small, but there's so many of them. So there's a lot of length to that. That's around the world 2.5 times. Okay, that's inside of every single one of our bodies is 60,000 miles of blood vessels. So literally that goes around the world 2.5 times, about 25,000 miles around the world. So that's inside of each of us. Okay, and so when I think about that, and I think about the incredible way that we actually control where blood goes in that 60,000 miles. That's the amazing part, right? It's not just that we have all these pipes that we can send blood in, but it's actually that our bodies can decide where to send more blood to or less blood to at a very sensitive level. I can't help but think of this verse here, which is one of the classic ones anatomy professors use, but I do still love it. For you created my inmost being, you knit me together in my mother's womb. I praise you because I'm fearfully and wonderfully made. Your works are wonderful. I know that full well. Yeah, so definitely remarkable. Just a little factoid about the human body for sure. So, okay. So today we're really just looking at kind of anatomy of the different blood vessels, okay? And then next class, we'll get into the physiology a little bit, looking at blood flow and pressure and how those things are regulated. How are we doing with the sign-in sheet? Does anybody not got it yet? Put your hand up if he hasn't made it to you. Where's it at? Send it right back there to Hannah. Looks like she needs it. And then over this way, Hannah. When you okay, thank you. All right, so a little bit of anatomy of the blood vessels, and really it's a lot of categorization of all these different blood vessels, okay? You guys have been studying these already in lab, and now we'll get to look at them in a little bit more detail here. Okay, so arteries always go away from the heart. Capillaries are in the middle between arteries and veins, right? And that's where all the business of the vascular system really happens, which is really the diffusion, right? Things can come out and in only really at the capillary level, okay? Veins are always going back towards the heart. And then there's this kind of other little category that we don't hear much about called anastomosis, okay, which is when these veins and arteries merge together, okay? Yeah, I think we need it right over here, yeah. Okay, yeah, when veins and arteries merge together, that's one example, but we also have arteries that merge together as well, okay? So the circle of Willis is an example of that. And did you guys talk about that one yet in lab, circle of Willis? Yeah, not yet, you'll get to it. I think next time we do the cardiovascular anatomy lab. But that's up in your brain, and it's where one artery comes up, and normally we go artery, and then it goes to capillaries, and it goes to veins, right? But in arterial anastomosis, it goes up, and then it meets up with another artery instead, and then continues to supply something. And the reason for that in the brain is because if one of those arteries gets blocked on one side, then actually the blood can come up the other way instead and still make it all the way around to that blockage on one side, where it's coming down the other way and can make it around the blockage on the other side. So we have this kind of backup system in very important areas like the brain. Okay, so that's what an arterial anastomosis looks like. We can also have an arterial venous anastomosis, which pretty much just means we skip the capillary bed. Right, so we go from artery to vein, and we skip the capillary bed in the middle. Okay, this is also known as a shunt. Okay, and we have these are very common in the body. Okay, so if a certain area of tissue doesn't need oxygen at that point or doesn't need extra blood flow, then it's just going to skip the capillaries and keep going. And then the venous ones, this is where two veins connect to each other, and these are actually pretty common for veins. Okay, so really just those four categories of blood vessels: arteries, capillaries, veins, and anastomoses. And then underneath those anastomoses, we've got 3 different types. Okay, questions on that so far? Okay, all right, so let's take a second. Go ahead and kind of just match those up, label these, okay, the ones we just talked about, and with the letters, okay? And then you can send in the answer to this question here on the app. All right, feel free to talk to the people around you if you're here in class. I'm not going to send you to your breakout rooms on Zoom for this one, but we will a little bit later. So just matching up those letters with the names there and then sending in the answer to that question on the app. All right, so send in your answer to the question on the app there, give you about 15 more seconds. So which one is to ensure blood gets to a certain tissue, even if an artery is blocked? So in figures and pictures like this, we can really only tell the difference between an artery and a vein because of the way that the artist colors it, right? It's not really, doesn't really look blue or red. Yeah, and then in the dissection specimens, they're colored with silicone, right? So you can tell the difference. Yeah, okay. Anybody else there with the question on the app? 66, yeah, it's probably about everybody. Okay, let's see what you said for that one. Okay, so got a few saying F and E. So I'm just typed it right in, nice. And then, yeah, about 40% of you saying A. Yeah, oh, there's, yeah, about 40% of you saying, oh yeah, 40% of you saying C, not A. There we go. Yeah, yeah, so C is definitely the right answer there. So C is arterial anastomosis. Okay, this is actually the circle of Willis here. You'll get into that, I think, in the next lab, not next week, but, or not this week, but the week after. Okay, so then which one is just the actual artery? That one would be B. Okay, the other side of that, the vein, that would be A. Capillary bed is in the middle of those two, F, right? Venous anastomosis. So you can see with the veins here, the veins are coming down, and they just kind of loop around, they connect again there, connect again there. So obviously it doesn't go artery, vein, or artery, capillary, vein, it's just kind of going vein to vein to vein. And that's, veins are just kind of tricky like that. They just connect in all kinds of different places with each other. That's just kind of how veins are. So that one is D there. And then the arterial venous shunt is E. Okay, so you can see if blood just shoots right through here and isn't allowed to go out here to these capillary beds, then no diffusion can happen. It just kind of skips over from the arteries to the veins. Okay. All right, any questions on those? Oh yeah, so this one was C. All right. Okay, so this is kind of our initial categorization. And then when we look at each of these different types of vessels, they'll have some similar features to them, okay? And the biggest similar feature is the different wall layers that they have, okay? So the most external one is called the tunica externa, okay? Also known as the adventitia. This is the most outer layer of these blood vessels. It's protective. It's mostly connective tissue. And then that connective tissue, it connects it to the rest of the body to kind of hold it in place. Okay, then there's the tunica media. This is the middle layer. This is where the muscle is, okay, the smooth muscle. The smooth muscle is responsible for constricting and relaxing to change the size of the blood vessel. Okay, so when this smooth muscle contracts, that leads to what's called vasoconstriction, which decreases the size of a vessel, and that decreases flow. Okay, relaxation, that leads to vasodilation and increased blood flow. Okay, and then the most internal layer is the tunica intima, or the interna. Okay, this is the inner layer, very smooth, so the blood can flow nicely down. The cells on the inside of those walls, known as endothelial cells, are very important for blood vessel and really full body health. And it's really one continuous layer of these cells through all 60,000 miles of blood vessels. It's just this one continuous layer, okay? And so again, those endothelial cells are what line this. And remember, this is one example of epithelial tissue, right? Epithelial tissue, yeah, this was one of the ones you had to know, the endothelial layers in the blood vessels. And this was an example of simple squamous epithelial tissue, just those single cells that are really flat, okay? And we'll talk more about this when we talk about cardiovascular disease at the very end of the semester. But endothelial dysfunction, when these cells start not working well, is really the first step in the progression of cardiovascular disease, which is the #1 thing that kills people in our country. And so these cells are really kind of at the root of that disease. Okay. All right, so we can just kind of walk through this one together here. So the vessel wall labeling, okay, so we can see with A there, that is the tunica intima that contain, we can see the endothelium there, that endothelial layer, that one little layer of flat cells, okay? And then from there, we get to the next layer, the tunica media, that would be the smooth muscle there. And depending on the blood vessel, it's gonna have a varied size of that smooth muscle, okay? And then C is our tunica externa. And the interesting one here, see there's a capillary, this one's artery, this one's vein, right? And capillary in the middle. Capillaries only have the tunica intima. They don't have the other layers, right? So they're very, very small. You can just see those little endothelial cells and the little basement membrane next to those. Okay. And then D is just a unique feature that we find in veins, which are valves. Okay. So it's a very low pressure system on the vein side of our vascular system. And so when blood passes one valve, we don't want it to take one step forward and two steps back, right? If it can kind of just flow backwards if gravity was pushing it that way. So instead, once it passes a valve, it stops there and it can't go back any further. Okay. Yeah, and let's do a quick little thing. You can do it on your own body to see if you can find a vein. Okay, the way this works is if you have veins that pop out like I do, then pretty much what you do is you take a finger here and kind of cut off your vein with one finger. Okay, and then you take your thumb and run it up like this, and that's pretty much pushing all the blood past those veins. It definitely doesn't work to hold it up like this, like I'm doing. I'm just trying to show you guys. You really want to hold it down if you're doing this. But yeah, if you kind of just pinch off your vein in one spot, and then you take your thumb and run it down, what you might see is once you're done moving your thumb, then you see blood come back to a certain point, but not all the way back to your finger. And you can see like a little bump there, and that's where a valve is. So you can play around with that a little bit more later, but that's the idea, is you're pushing the blood up past the valve, and then it comes back to that valve again, but it won't come all the way back to that finger that you have pinched off there. So just a fun little trick to find your vein, your valves. Okay. All right. So when we look at the whole vascular system and all of our blood, about over half of our blood is found in our veins at any given time, in our systemic veins, 55% of it. Okay. So that's really one of the functions of our veins, our systemic veins, is to be a place where blood just kind of pools, like a kind of this reservoir for blood. Okay. But yeah, so let's start off first talking about the arteries, though, the arterial side of this. Okay, so our arteries get smaller as they move away from the heart. Okay, because blood's going from the heart through arteries and out. Biggest artery in the body is right there at the heart, right, the aorta, and then it just gets smaller and smaller and smaller as you keep going. Okay, in arteries, the tunica media is the biggest, the thickest layer. Okay, that gives it strength, but that also gives it a lot of smooth muscle so it can change size. Arteries are both elastic and contractile. Okay, so they're elastic, meaning that when we stretch them, they bounce back. Okay, and that's exactly what happens every time your heart beats. All right, so you guys have probably all found your pulse before, but if you haven't, you should try to find it with two fingers. Kind of just go right down from your thumb. Right, remember that old pisiform bone we found last semester, right? Go right inferior to that, okay, really medial if you're like this, and put your two fingers there. You should be able to feel your radial pulse, and that little pulse you're feeling is literally just a stretch of your artery walls that started all the way back at your heart. Right, it's that same exact stretch. So your heart pumped, and it put so much pressure on your aorta that it stretched out the walls. And then that same little pressure wave went through the whole vascular system until it made it there to your fingers, right? And so that's what a pulse is just that same pressure stretch, right? And the kind of cool part about that is it not only stretches and withstands that pressure from the heart, but it also bounces back. So it kind of pushes the blood from behind and keeps it going. And that's one of the functions of the arteries with that stretch and bounce back, is it keeps the speed up and keeps your blood pressure up by pushing it along after it stretches. Okay, and so that's the elastic part, but the contractile part is really controlled primarily by the sympathetic nervous system. Okay, and so the more sympathetic nervous system activity we have, then the more constriction we have. Okay, the more that shrinks them down. The more, the less sympathetic nervous system activity we have, then the more dilation we have. The more that opens them up. Okay. All right. Any questions so far? Okay. All right. So different artery types. The largest ones are called the elastic arteries, also known as conducting arteries. They have a very big lumen. That just means the inside of it, so more blood can get through. The main function of this type is to get a lot of blood somewhere fast, right? So this would be like your aorta, all those big arteries up that branch off of your aorta. Okay, so the aorta and the major branches, pulmonary trunk. Okay, they have those same layers. The externa is somewhat thin because it's not really that interconnected. They're so thick they don't need connective tissue to hold themselves together. The media is extremely thick. Okay, mainly the elastic portion. Okay, to give it strength, so it doesn't burst under lots of pressure. Okay, that's the main reason there. And then to give a lot of bounce back too, right? Because this is the most important place to keep blood pressure up, is right there where the pressure begins, is at the heart. Okay, and then the intima is relatively thick as well. Again, need that support to withstand that pressure. Okay, so those are the elastic arteries. We've also got muscular arteries, okay, also known as distributing arteries. These are kind of the intermediate ones. They're kind of medium in size. Okay, their job is to kind of, just like it sounds, distribute blood to different parts, different regions of the body. Okay, these are most of the name ones that you had to study and you're studying this week. Okay, and the layers kind of got those same layers there. Extern is very thick in this case. Media is also very thick because we can change the size of these blood vessels a lot. And then lastly, we get to the arterioles, which are the smallest ones. These are very small right before we get into capillaries. Okay, function of these is to regulate how much blood flow goes to the capillaries. Okay, but then secondly, this is where most of our blood pressure regulation occurs in our body. Okay, so when someone someone gets a bee sting and they go into anaphylactic shock, right? Their blood pressure drops drastically because they get lots of vasodilation. These vessels here, these arterioles, are the ones that have the most impact on that. Partially it's because there's so many of them, because they're so small, and there's a lot of blood in all of these together. But then they also just have a lot of room to change size because they have so much smooth muscle. All right. Okay. So yeah, take a second. Go ahead and label these arteries here, and you can answer this question down here at the bottom. Again, you can check with your neighbor here in class, if you're in class. Smooth it. Let's see. What did I say there? Yeah, a lot of strength and then the bounce back. Yeah, it wasn't smooth, it was strength. Yeah, so.
We're just naming what type of arteries they are. Are we naming the name?
You're just labeling them, yeah. Yeah, just the type, yeah, not the names. Oh. Yeah. So just make sure you're answering that question there in the bottom. Where is the site of most blood flow control? We could also change that to blood pressure control. All right, so let's walk through this together here. So if we're starting at the heart, the biggest ones right up to the heart are what? What are those? Yell that out. Yeah, so those big elastic or conducting arteries, okay? And then the next one down are muscular or distributing arteries, right? And then we get to these little tiny ones that are leading right to the capillary beds, right? So those are going to be the arterioles, also known as resistance vessels. Okay, and then we've got two different ways to kind of, what's known as a meta-arterial, okay, which is leading right to, right into that arteriovenous anastomosis, okay? So it's the same idea as that arteriovenous anastomosis, that shunt, where it skips the capillaries. That's what another name for that is, meta-arterial. Okay, so let's see what you guys said for the question here. All right, yeah, so about 60% of you are saying C, which is correct. Yeah, that should be the arterioles, right? So the arterioles are the ones where we really control blood pressure the most, which help us control blood flow as well. And again, it's because they have smooth muscle, so we can constrict them and dilate them. But they're also small, and there's a lot of them, right? So we have a lot of our blood is in these, right? So that means by changing the size of those, we change the pressure quite a bit. We're impacting a lot of the blood. Okay. All right. Any questions on those? Okay. So those are arteries. Let's talk about the ones in the middle now. So we're going from artery to capillary, and then we'll go to the veins last. Okay. So the capillary system is the location of this, again, is between the arteries and veins, but more specifically between the arterioles, which we've talked about. We haven't talked about the equivalent of those arterioles on the vein side, but those are called venules. Okay, so the function of the capillaries is this is where all the exchange happens, right? This is really where the most important part of the vascular system occurs, which is diffusion of nutrients, diffusion of gases, diffusion of waste products. Okay, the structure of these little guys is endothelium only, right? So just that intima layer is all that's here. Okay, capillary beds have those meta-arterioles, like I just showed in the other one, which are just the more specific name for that arteriovenous shunt, right? That's part of the artery, just connects to the vein and skips the capillary bed. Okay, but then the capillaries themselves are usually somewhere, each capillary bed is somewhere around 10 to 100 tiny little vessels. And literally a single red blood cell fits through these at a time. So it's like a single file line of red blood cells going through these, okay? So extremely small, microscopic, right? And the cool thing about capillaries is we can open and close them if we need them or not, okay? So there's these little sphincters at the start of the capillaries. And if we open them, then that allows blood in for exchange of nutrients. If they're closed, then it just skips, it goes through that meta-arterial, through the anastomosis, and no exchange happens. Okay. All right. So then let's look at a couple of different types of capillaries. Okay. So the different types we've got here, they're really just three types: continuous, fenestrated, and sinusoidal capillaries. Okay. The continuous ones are found. There's a few locations given for each of these. Okay. So we find these in the brain and the CNS. They're part of this blood-brain barrier. Okay. We find these in the skin, in the muscle. Okay. These are very important for allowing diffusion to happen, but also creating a seal. Okay, not much can leak through these capillaries. These are the least leaky type of capillaries of the three. Okay, because there's a continuous wall. Okay, the fenestrated ones actually have holes in the cells themselves, little pores. Okay, and this allows for a little bit more leakage, a little bit more diffusion, more permeability. Okay, we have these in places where we do a lot of filtration and absorption. Okay, so those would be the kidneys, that filters our blood, the endocrine glands, that's how we a lot of times how we can get hormones out of endocrine glands is to leak them out through these fenestrated capillaries. And then intestines, helps us absorb nutrients. Okay, so places where things are having to either enter the blood or leave the blood commonly, like the kidneys, the endocrine glands, and the intestines, is where we're going to find these. Okay, and then lastly, the sinusoidal capillaries. These are the leakiest types. They have holes in the cells, and they also have big gaps between the cells too. Okay, so these are places, we find these in places that actually have something to do with either forming the blood, modifying the blood, doing something to actual blood cells. So instead of just little nutrients or hormones leaking through the holes, we actually need the whole blood cells to be able to leak in and out of the capillaries. And so that's why we have these larger holes in the sinusoidal ones. Okay. All right, so this one kind of walks us through. I can just walk you through this here. So the 3 different types, right? So we can see B definitely looks like the least leaky, right? Here we can see these holes, these little pores, not much gaps between them. And then this one has big holes and big gaps between them, right? So A is the sinusoidal ones, B is continuous, and then C is the fenestrated ones. Okay, and the most common one for sure is going to be B, the continuous ones. Okay, the other 2 are kind of more specific for very specific functions in certain parts of the body. Okay. All right, any questions about any of that? We got arteries so far, we got capillaries. We'll move into veins next. Okay, and this is just kind of showing how that meta-arterial works and how those precapillary sphincters work. So these little things here are representing the smooth muscle that can close or open and allow blood to go into the capillaries. And so when the capillary beds are closed, or when this smooth muscle is contracted, it just skips right through, right? But then when these open up, these relax, then the blood can go through the capillaries first before it comes back this way. Okay, so lastly to the venous system. These increase in size as they move towards the heart, right? Which is really the same as the arteries. It's just the fact that the blood is flowing a different way, right? It's flowing towards the heart and veins instead of away from the heart, okay? So the function of these is number 1, to get blood back to the heart. And number 2, allow for kind of this blood pooling or this blood reservoir for us, okay? And then under times of stress or under times where we're exercising, we need more blood in the general circulation. We can mobilize that blood pool by constricting the veins, which sends you got that 55% of the blood out there. So that sends some of that blood back into general circulation. And that can help us increase our blood going back to the heart. This is exactly one of the ways that we increase that end diastolic volume, like we were just talking about in the heart. Remember, right? When we're starting to exercise, our end diastolic volume starts going up so we can increase our stroke volume, right? One of the main ways we do that is by sending more blood, getting it out of the veins and pushing it back, okay? One of the ways that the veins work together with our skeletal muscle is something called the muscle pump, okay? And this is a nice image that shows how that works. Veins are commonly found kind of between muscles or between muscles in the skin or between muscles in a bone or something like that. So when the muscles contract, they smash the vein down, right? And so we can see that here, there's when the veins open, muscle contracts, it smashes that down, right? So the blood's gonna try to squirt both ways when that happens, right? But luckily we got these valves, right? Because the blood that went this way, that's fine, it's gonna go right towards the heart, okay? But then the blood that's gonna try to go this way, it's gonna hit that valve, that's gonna make a U-turn and head back towards the heart, right? So as soon as you start exercising, this is another way that we increase our end diastolic volume, because you start contracting a bunch of muscle, right? So all of a sudden that blood is pumping back towards your heart. Without the valves, this wouldn't really do much for us, right? Blood would just be squirting both directions. So the valves are incredibly important for this. Okay. All right, let me show you this little video. Part of the research that I do, and I did my dissertation in my PhD program, was looking at the brachial artery in people with an ultrasound machine. Okay, so this right here is the brachial artery of a subject, but then sometimes you would get the brachial vein too, which is right below it. Okay, so that's right here. the brachial veins right there. And every once in a while you would get a really good view of a valve. In this case you can't. So watch where that little circle is when I play this, and you'll see this little like kind of leaflet thing flopping up and down, and that's the valve. You see those? You can actually see both little leaflets of the valve kind of flopping up and down. And you can see every time the heart beats, that's what opens the valve, because actually the artery right next to it pushes out on the vein a little bit and smashes it down and pushes that blood flow through. So you can see those the little valve leaflets kind of flopping up and down. So that's a valve right in the brachial vein. Okay. All right. So the vein wall structure, the layers are thinner than the arteries for sure. The thickest layer is the external one, the one that kind of holds it to other parts of the body. Okay, that's the externa, the tunica intima. The valves are actually just formed as extensions of those endothelial cells. Okay, so these help us fight against gravity. Okay, because when you guys have probably seen on airplanes, and sometimes they'll recommend for you to, if you're on like a long flight, for you to stand up and do some little like heel raises and things like that, right? That's because your blood is just pooling down in your feet, right? And so just by contracting your calf muscle a little bit, that pushes some blood up, okay? And just maybe one pump gets some blood up above one valve, and then it kind of sits back down on that valve, right? And then you contract again, and it goes up another step, right? And that's really where those valves are super helpful, because otherwise it would just fall right back down, right? So, okay. The venules are the smallest ones on the other side of the capillary beds. Venous sinuses are these really flat veins that we talked about these in the heart. This is part of where the blood dumps from those coronary arteries back to the heart to get back to the right atrium. Okay, also up in the brain. Okay, one pathology that you can have with veins are called varicose veins. This is when the valves aren't working well. And so what happens is so much blood pools out in those veins as they get kind of stretched out, okay? If you've ever studied a little bit geography, then you know that the oldest rivers are the ones that curve around the most. It's called meandering, right? The more a river meanders, the older it is. It's kind of the same idea with these veins. The more like stress you have on these veins, the more they meander around like that. And you can see that in these varicose veins. These can be pretty painful. And obviously unsightly, right? Yeah, some of the causes here, some different things that lead to it, but there's definitely a genetic component to these. Okay, so let's take the last few minutes here. I'll send you guys on Zoom to your breakout rooms. Just go ahead and walk through this activity here. So what are the functions of each of these vessels above, okay? And then I want you to go through and answer these questions here together, okay? And then the one you'll send in on your app is this one here again. Okay, go ahead. We'll take a few minutes to do this, and we'll wrap up with that. All right, so you're sending in that question on the app there. Where does the most blood pressure control occur? All right, everybody should be coming back there on Zoom. Go ahead and send in your answer there if you haven't yet. So where does the most blood pressure control occur? Almost got everybody. Okay, so most people are saying C now. Good, so that's absolutely right. Those arterioles is the place the most blood pressure control occurs. So that is C. Okay, so we can look at each of these. So those elastic arteries, their function is conduct blood under high pressure organs, right? The muscular arteries control blood flow, regulate blood pressure. Arterioles control blood flow to the tissues, right? This is what gets us right to the capillary beds. We could add this one, very important for blood pressure control. And then the venules, this is just kind of that last part, the first part of the venous side after the capillaries and the veins returning blood to the heart. So where does diffusion occur? What's the name of those blood vessels where diffusion occurs? Guys in class, girls too. Say it out loud. It's not up there, right? It's not one of those. It's capillaries, right? Capillaries where diffusion occurs. Where's the most blood found at any given point? That's going to be the veins. Okay, those guys. Which one is the most abundant? It's going to be somewhere between arterioles and venules out of the ones you can see there. If capillaries were involved, it would definitely be capillaries. Okay, it's always just the smallest one is going to be the most abundant, right? So capillaries are definitely the smallest. We got the most of those. If that's not one of the options, then it's one of the two on either side of the capillaries, which would be the arterioles or the venules. Okay. All right, guys. That's it for today. We're done. Have a great rest of your day. And we'll see you guys on Wednesday on Zoom. I'll see you guys on Zoom back here in class.
Dr. Sawyer, I don't know if you can hear me.
Oh, yeah, hold on one sec, Caleb. Let me get you off the speaker. All right, Caleb, you there?
Yeah, I'm here.
Hey.
I had a question on the vein wall structure. I went to the
Okay.
bathroom and I missed the fill-ins on that page.
Oh, gotcha. Oh, that's okay. I can just put that up there for you.
Thank you.
Yeah, for sure. Hey, Jojo, do you have a question?
See you Wednesday.
All right, Caleb, see ya. Perfect, Jojo, thank you. You got it now? Have a good day.